This is a hemoglobin molecule, and the hemoglobin molecule is made of 4 polypeptide chains. We have 2 alpha and 2 beta polypeptide chains, and in the center of each polypeptide chain we have a little heme group. And this heme group, even though it's green here, this is the pigment that makes the hemoglobin molecule red. And so here's a heme group, heme group, heme group, heme group. In the center of each heme group we have an iron atom. And this iron atom, see the iron atom here, this is where the oxygen molecule attaches in each hemoglobin molecule. So we've got oxygen would attach here, 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 and here. So each hemoglobin molecule can hold 4 oxygen molecules. And each red blood cell can hold millions of hemoglobin molecules, like 250 million. So each red blood cell can hold 1 billion oxygen molecules. That's a lot of oxygen to be carrying around inside of it. So these hemoglobin molecules are made of 4 polypeptide chains, and each polypeptide chain has a heme group in it, and each heme group has an iron atom in the center of it. See how important iron is to our diet? You can't make hemoglobin without iron. And iron is where the oxygen attaches. And so that's the only place it attaches. Inside the red blood cells, oxygen is being carried, attached to the hemoglobin molecule, attached to the center of these little heme groups here. Now sometimes carbon dioxide is carried inside of the hemoglobin molecule, but not often. Usually carbon dioxide is dissolved in the plasma when it's carried in the blood. So it doesn't compete for space with oxygen, because we wouldn't want carbon dioxide and oxygen being competing for space inside of our red blood cells. That wouldn't be very good. But if carbon dioxide does get carried inside your red blood cells, when it does get carried, it attaches to the polypeptide chains. It does not attach to our heme group here. So it does not compete for space with the oxygen molecule. There is a molecule that does compete for space, and that would be carbon monoxide. If there were an oxygen molecule trying to attach here and a carbon monoxide molecule trying to attach here, carbon monoxide would win. It has a higher affinity for this iron atom than oxygen. Alright, so we don't want carbon monoxide anywhere near us because it will win. Now, when oxygen attaches, let me show you what would happen. Get to the next slide. When oxygen attaches to hemoglobin, then we get a molecule called oxyhemoglobin, and it changes the structure of that hemoglobin molecule. It makes the blood actually turn a bright red. This is oxyhemoglobin right here. Oxyhemoglobin occurs in the lungs. That's where we're going to pick up oxygen. And then we're going to take that oxyhemoglobin, we're going to take our red blood cells to the tissues where oxygen unloading is going to occur. And oxygen unloading naturally occurs. It's not like the oxygen just decides to leave the red blood cells because it feels like it. There's no oxygen in the tissues. So it's just going to be a natural diffusion of oxygen from the red blood cells into the tissues. So when oxygen leaves the hemoglobin, then we call the hemoglobin reduced. We say it's in the reduced state, and we call that hemoglobin now deoxyhemoglobin. And this is what deoxyhemoglobin looks like. It's a darker red color. This is deoxyhemoglobin right here. When we're in the reduced state, when hemoglobin is in the reduced state, that's when it will pick up carbon dioxide. And this is going to occur in the tissues because that's where carbon dioxide is. It's being produced by our tissues when they're making ATP, doing cellular respiration. So carbon dioxide is going to be picked up by hemoglobin in the reduced state, and we're going to make a molecule called carbaminohemoglobin. And this is what that looks like. Now, red blood cells only live so long, and they're only going to live 3 to 4 months because they're anucleate and they can't undergo mitosis. The structural net that makes up the plasma membrane, the internal facing of the plasma membrane, is going to just get old, and they're going to start to become just brittle. And as they pass through the spleen, which is our red blood cell graveyard, the spleen is going to grab onto those old, dead, dying red blood cells, and they're going to munch on them. Basically, they're going to phagocytize them. But we don't just throw away our old red blood cells. We're going to save all the hemoglobin inside of them. And what's going to happen is the globin portion, that's the easiest part. The globin is just, they're polypeptide chains. So we're going to take polypeptide chains and break them down into their amino acids. And those amino acids will be recycled. The harder part of this is to figure out what happens to the heme. The heme is a red pigment. We're going to take the iron out of that pigment and we're going to store it inside of our cells as ferritin or hemosiderin. But the pigment part, the pure pigment, the red pigment, is going to be broken down into a yellow pigment, a yellow-greenish pigment called bilirubin. And that bilirubin will go through the blood until it's picked up by the liver, and the liver is going to secrete it into a substance that it makes. The liver secretes a substance called bile, and that bile is going to be used in our digestive system. What bile does is it's going to physically break down fats. And so the liver will take the bilirubin out of the blood and put it into bile, and it's going to secrete that bile into our small intestines. And as the bile gets squirted onto our food and goes through our food, and our food goes through our small intestines and into our large intestines, as that yellow pigment goes through there, it's going to be converted into a brown pigment called stercobilin. And so that's what gives our feces the brown color that it is. So we go from a red heme to a yellow bilirubin to a brown stercobilin color as the heme is broken down. Now, if anything's wrong with our liver, our pancreas, or our bile duct, or any of the tubing around that area, we can have a backup of this bilirubin in our blood. And if the liver can't take it out and excrete it into the bile, then the bilirubin backs up into our skin and it is deposited into our skin, into the adipose layer, and also it can be in the stratum corneum. And what will happen is we get jaundiced, and so it shows you the yellow coloring of the eyes. You can see yellow coloring in the eyes here, and not the best picture, but here shows you yellowing of the skin. And hopefully if your liver gets fixed, then the problem goes away. But in newborn babies, how you resolve this issue is phototherapy. You can just lay your baby in a sunny window, or UV radiation, like this baby is doing UV radiation. You just put it over their trunk and you just lay them in light and the UV radiation breaks up the bilirubin. So here's the yellow baby before and then after. And that's it.